Welcome, my beautiful people. So this is our fourth part of our series of biology paper 2 with random questions from different past papers. So this video will focus on questions under the topic of genetics. So we have the first question here. Figure 5.1 below shows pedigree diagrams for two families A and B, outlining inheritance of sex-linked disease called hemophilia. So we are looking at a sex-linked disease known as hemophilia. So there are also other types of diseases that we can consider sex-linked, such as we may have red-green color blindness, also sickle cell anemia. So whichever disease or question comes under these three types of diseases here, the concept is the same. So the same way we are going to answer for hemophilia can be handled with red-green color blindness or sickle cell anemia. So the only thing that you may encounter is the use of letters. So under hemophilia, we use small letter H and capital letter H. Then for red-green color blindness, they may use R. So whichever letter is used, the concept remains the same. So if you look at genetics, genetics is a science or study of characteristics passing from one generation to the next generation, or the passing of characteristics from the parents to the offspring. So during fertilization, that is when the characteristics are passed on, because there is the fusion of the sperm cell with the ovum from the female. So during that fusion, then the characteristics also are being shared. Now, away from there, when you are looking at sex-linked disease, there are some points that you need to know. First, let's understand the key here. So for the key, you may have a circle which is unshaded, or a box which is unshaded. For a circle which is unshaded, it means the woman, that is a normal female. Now, the unshaded circle can also mean that the female is a carrier. So when you say someone is a carrier, that person is okay, but has a defect that can be seen in our offspring. Then for a man or a male, it's always like normal if it's not shaded. There is nothing like a carrier. Men do not act as carriers. It's either they are sick or they are 100% okay. Now for a female who is hemophilic, the circle is shaded. For a man, the box is shaded. Now, let's come up with the genotypes concerning these diseases. So we use hemophilia because that's the question that we are using. But remember, the concept is applied to all these three types of diseases. So I'm going to begin with a woman. So let's say a woman. Number one, let's say if the woman is a carrier. So how do you write the genotype for a woman who is a carrier? So remember, for a woman, we are going to have XX chromosome. Then, when she's a carrier, they are going to have a capital letter H and a small letter h. So carrier is indicated by a mixture of a capital letter and a small letter. Then, what if the woman is 100% normal? So for this case, you're going to have two capital letter H's like this. So whenever they say a woman is 100% normal or pure, then you use the same letter as the HH. Now, what if the woman is a patient? So a patient woman will have small letter h, two small letter h. So these are the three types of genotypes when we are looking at sex-linked diseases. Remember I said what we have written here can be applied to all these three sicknesses. So the only difference is letter. If you are using, let's say, R, so for a carrier, capital letter R and small letter r. For normal, two capital letter R's. Then for a defect, two small letter r's. Now what of the man? So for a man, if the man is normal, so the chromosomes for a man are X and Y. Then Y has no space, it is smaller in size, so therefore Y do not carry the disease. So do not put any letter on letter Y. So since this person is 100% okay, you only write capital letter H on X. Y do not put anything. So what if the man is a patient? So if the man is the patient, then you put small letter H on X. So this is now how you are supposed to come up with the genotypes. Now, let's now look at the first question, A, Roman number 1, from figure 5.1. Which family has a parent who is a carrier for hemophilia? So, to identify the family that has a parent who is a carrier, first we need to look at the offspring. Do we have any offspring who is hemophilic? Let's look at the offsprings from the both families. So let's look at family B. So family B, we can see that all the polygons here were unshaded. So it means there is none. So among these offsprings, there is none who is a patient. Now let's check for family A. So family A, we can see that here we have a box which is shaded. It means that there is a son who is sick. And since there is a son who is sick, then one of the two parents is a carrier, which is a woman, because a man cannot be a carrier. So we can say family A. Give a reason for your answer in A1 above. We can say because they had a son who was hemophilic. So the defect that was in the mother appeared in the son. Then for B1, using the symbols XH and X small letter h, state the genotypes for offsprings H and J if J is a carrier for hemophilia. So here, now let's look at H. So H is shaded and it's a box. It means the man is sick. When the man is sick, you are supposed to use this. So you write like this. Now for J, we are told it's a carrier. So when the J is the carrier, so for a carrier, we say it's a combination of letters, capital and small letter. So you put capital letter H and small letter h like that. Then using a genetic diagram, show whether the offspring would be hemophilic or normal when H and J are crossed. So we have H, then we have J. If you want, you can say man, woman, or male, female, whatever that comes into your mind is still okay. So I'm going to begin with parental phenotype. So parental phenotype for H, we know that H is sick, so we just say patient. J, carrier. Then you move on to parental genotype. So parental genotype for H, this. Then we excuse me, we move on to parental gametes. So I'm just going to split these. Then we can start crossing. So this one goes with this one. So always start with the capital letter. So X with capital letter H, X with small letter h, like this. So it means there is a chance of having a girl child who is a carrier. So we have X, X, then we have small letter h, small letter h. So there is a chance for this couple of having a daughter who is a patient. So there is a chance of having a son who is 100% okay. So there is a chance of having a son who is a patient. So a genetic diagram just gives you possibilities of something to happen or not to happen. So here you can now say genotype of F1, so first filial generation. So for carrier, we have 1 carrier, we have 2, so this is a patient, and this is a patient. So for a patient, we have 2 possibilities. For 100% normal, we have 1. So if they ask you to maybe write these as like percentages or a chance, so let's say like if they say what is the percentage, what is the chance of having a normal child? So for a normal child, you can just say normal is equal to, for a normal 100%, we only have 1. So you have to say 1 over 4 multiplied by 100% gives you 25%. So for every fertilization, 25% of that fertilization can result in having a son who is 100% normal. What of the carrier? So the carrier is also 1. So it's 1 over 4 multiplied by 100% is also giving us 25%. So what of our patient? So for patient, it's 2 over 4. As we can see, we have 1, 2 times 100%. So 2 over 4 is same as half. So half of 100% is 50%. So 50% of that fertilization can result in giving us a child who is hemophiliac. Right, so let's move on to question number 2 and blood groups. So Mrs. Momba delivered a baby girl in a certain hospital. Mrs. Momba has blood group O and the father of the girl has blood group AB. Mrs. Momba sues the hospital for giving her a wrong baby whose blood group was O. Explain using a genetic diagram why Mrs. Momba would win or lose. So if this lady had taken a step of taking the hospital to the courts in charging of her giving the wrong baby. So now we want to use a genetic diagram to explain if this lady would win or lose the case. Now before we dive into coming up with this genetic diagram, first let's have some notes of blood groups. So for blood groups, you need to remember words like homozygous and heterozygous. So homozygous, it means same letters. Heterozygous, different letters. So we'll begin with number 1, blood group A. So depending on the question, blood group A can be heterozygous or homozygous. So if blood group A is homozygous, then you are supposed to use the same letters. And AA cannot produce blood group O. It's not possible. For blood group O to be produced in an offspring, at least in each genotype, there should be an existence of O. Now, what of heterozygous for blood group A? You say AO. So even if you have AO, you don't have to read this as blood group AO. It's blood group A. Since O is recessive, A is dominant. So O cannot express itself when it is in a combination with B or A. Let's move on to blood group B. So for blood group B, it's same procedure with O. So homo, if they say homo for blood group B, it's BB. BB cannot produce O. Then what of its hetero? So if it's hetero, it's BO. Now what of blood group AB? So for blood group AB, the genotype is always AB. There is nothing like hetero. So why is this AB? The reason is simple. A is dominant, so is B. So a case where we have two characteristics that are all dominant, they are trying to express themselves at once, gives us what we call the codominance. So the situation like this one is called the codominance. A codominance can also exist in like colors, where you have black and white exist at same time. Now for this point, we are saying A is dominant, B is dominant, so they are going to express themselves at once. Thus, AB is codominance. Now what of blood group O? Blood group O always exists in homozygous. There is nothing like hetero. For O is always OO. So if they say an individual has blood group O, it means we are going to have OO. Now, what you need to take note of this is that homozygous AA, BB, and AB cannot have blood group O. So the point is, if one of the parents has AA or BB or AB, there is no chance of having a child with blood group O. So it doesn't matter whether the other has OO, AO, BO, as long as in one of the two there is no O, then for that couple it's not possible. So let's see, even before we answer the question, we are told this Mrs. Mumba has blood group O. It means the genotype for Mrs. Mumba is OO. Now for the father, it's AB. So from the AB, we don't have O. So this couple cannot have blood group O. It's not possible because there is no O in the father. So to summarize our point, we are saying for us to have O, there should be at least one O in each. For example, OO with AO, yes, it's possible because there is at least one O. OO with BO, it's possible. OO with OO, it's possible. AO with AO, it's possible. BO with BO, it's possible. What else? AO with BO, it's possible. So the point is, at least there should be an O in each genotype for the couple to have a child with blood group O. So when you take that note, it is going to assist you with these questions. So we'll start with Mr. Mumba. Then here we have Mrs. Mumba. As usual, you have parental genotype. Sorry, let's begin with phenotype. So Mr. Mumba, we said, so for phenotype here, you just have to state a blood group. So blood group AB here, blood group O. We'll move on to parental genotype. So here we are going to have AB OO. Parental gametes. So we split the letters now. Now we can okay, so here blood group A, so blood group again A, blood group B, blood group B. So as you can see, there is no chance for this couple to have blood group O. So now, since we've proved, so we can say Mrs. Mumba would win the case because there was no chance. There was no chance of having blood group O. Or if want, we can say because AB cannot produce O. Then question B, what could have been the correct genotype of the actual father? So it means this lady lied. She accused the husband of being the father. In actual sense, the paternity is somebody's. So the correct genotype can be AO, at least there is O, BO, OOO. So one of the three is correct. Right, so we move on to question number 3. So here we are dealing with now skin color. A pure breeding black bull. So whenever you just hear the word pure, it means homozygous. So homozygous can be as same two small letters. For example, BB is homozygous, or capital letter BB is also homozygous. So homozygous, same letters. So word pure, it means homozygous. So was crossed with a pure breeding red cow. So we have two animals that were in homozygous situation. Now all the resulting offspring were black. So remember, the bull was black and it was pure. The cow was red and it was pure. But the offspring, all the offspring took the color of their father, which means that the color of their mother was shadowed. It was ignored. It couldn't express itself because it didn't have that much strength to be released on the outside feature. So we can say red is recessive. So whenever a certain characteristic cannot be seen outwardly, then that is recessive. If red is recessive, then black is dominant since it appeared. Now using letter B or B for alleles, and remember dominant is shown by capital letter, recessive is by small letter. So since the cow is recessive and was pure, so the genotype is BB. Then the bull, the genotype is capital letter B, another capital letter B, since these two were pure. So if you can just understand this, then this question cannot be an issue. So which allele was dominant for skin color? So allele, they just mean one letter. So the one that was dominant is, you just write capital letter B. What was the genotype for parent black bull? Remember, it's capital letter B, another capital letter B. Why? Because it was pure, so it was homozygous. Using a genetic diagram, show the resulting offspring if the offspring black bull was crossed with the parent red cow. So we've got a bull here and a cow. So as usual, parental phenotype. So parental phenotype, here we can just say black. Here we have to say red. Number 2, parental genotype, black capital letter B, another capital letter B, small letter b, another small letter b. Then parental gametes, you split these letters. So this offspring was a black color because the capital letter B is dominant. So genotype of F1. So as you can see, it's simple. So suggest what could happen to the pure breeding black bull or red cow to cause them to produce a black and white offspring. So by crossing them with a pure white, so it can be white bull or white cow. Okay, so let's have another one with colors, but this time sure they are going to be a new term that we are going to discuss. So two farmers, one with a pure breeding black bull and the other one with a pure breeding white cow. So remember, pure simply means homo, same letters. Then even the white cow is homo. So black bull, we are going to have capital letter B, another capital letter B. Then for white, it's pure capital letter. Okay, for this one, we don't know whether we are going to use capital letter or small letter. So what we need to know is to understand the nature of their offspring. So the black bull was crossed with a white cow, and all the resulting offspring had a coat color called roan. So whenever you hear roan, it means it's a mixture of colors. So mixture of colors. If we say mixture of colors, it means all the two colors were expressed at once. They were seen. So it was like black, white, just like a camouflage. Now, if that is the case, it means the two colors were dominant. So since they are dominant, even for white, we need to use two capital letter W's. Now, using B for the allele for black color and W for the allele for white color, show the genotype of the offspring. So, we are going to have capital letter B from the bull, capital letter W from the cow. So, this is the genotype for the offspring. It's a combination of two capital letters since they are all dominant. Now, I explain why neither the bull nor the cow had their coat color. So, this question is like, explain why the offsprings did not take one color. The offsprings were not all black, or they were not all white, but they had a combination. So the reason is simple here. You can just say because the black and white colors were all dominant. And remember, this is called a dominancy. It's codominance. Codominance. So a codominance is where two different characteristics expresses themselves at once. Now, using a genetic diagram, predict the chances of the second filial offspring having a roan coat color when the parent, black bull, so black bull, was crossed with one of the roan offspring. So let's just say offspring. So parental phenotype. So here we have black color. Here we have roan. So roan, remember, white and black in our case. Then parental genotype. So for this one we have capital letter B, another capital letter B, roan. It's a combination. Parental gametes. We split the letters. Then the first cross. So homozygous for black. BW roan. Homozygous for black. Roan. Genotype of F1. This should be F2 since we have used F1. So as we can see, this is how we can handle a question like this. So if you want more of these questions where we can now have a discussion, where I can take you at your own pace, guys, so you can just contact me, join my online classes, so let us revise together. We are trying to have an intensive revision. So come join me. My WhatsApp line is this number 0962 89 58 04. So that's it for this video. Have a nice one.